Hello everyone, today we will be talking about Next sequencing system by Illumina. It is the first and only high-throughput desktop sequencer to offer exome, transcriptome, and a whole genome sequencing on demand. Here you see two systems. One on the left, the NextSeq 550, which is our older model, and on the right, the NextSequence 1000 over 2000 systems, which are the newer models, which have slight differences. Here for the working principle of Illumina, SBS, we go over a couple steps, such as sample preparation, cluster generation, sequencing by synthesis, and data capture and analysis. The first step we will be talking about is sample preparation, which has DNA input requirements. Typically, these DNA input requirements are between 1 nanogram and 1 microgram of high-quality DNA, which also means we need a base pair of 200 to 500 base pairs. For the RNA requirements, we at least need the range of 10 nanograms to 1 microgram, which also indicates we're going to need a RNA integrity number, which is above the number 7, which also produces around 200 base pairs. For this slide of specifications, I'll be telling you about DNA fragmentation. This is when you get a DNA strand and break it into fragments. This can lead us to now use single-end and pair-end fragments. Single-end uses an adapter on one side and reads one part of the sequence, whereas pair-end has two adapters, one on the top and one on the bottom. This reads both ways. The next step of SBS is cluster generation. As we see, we use our prepared fragments and place these onto a glass slide called the flow cell plate. After having these fragments on there, it will cause DNA bridge amplification, where each DNA fragment will bind to oligonucleotides in the flow state and generate thousands of original DNA into small clusters. The goal of this cluster generation is to strengthen the signal of fluorescent group bases so it is easily detectable and easily seen when we try to find the specific DNA we need to notice. The next step we will be going over is sequencing by synthesis. This sequencing is performed by one base at a time, using our enzyme called DNA polymerase, which incorporates fluorescent labeling nucleotides such as A, T, C, and G. Each nucleotide has a fluorescent dye attached to it. The nucleotide compete for addition to growing the chain. After each cycle, a light source excites this nucleotide, causing it to emit a characteristic fluorescent signal, as you've seen. The fourth step we'll be talking about is data capture and analysis. After each step, the system takes high-resolution images of the flow cell. It uses the color and position of this fluorescent signal to identify the nucleotide which is added. These images are processed and generate the sequencing data using algorithms that interpret the intensities of the signals from the flow cell. Next, we're going to be talking about the runtime of these systems, of the NextSequence system. This can depend on multiple factors, such as read length, single-end versus pair-end sequences, and the flow type. In short, for newer models, a single-end sequence of 100 base pairs would probably take around 13 hours. Meanwhile, the pair-end sequence of 100 base pairs would around take 20 hours to sequence entirely. Speaking about these machines, I will be going over the features that they also include, such as they are flexible with multiple read lengths and output options. They are simple and easily usable interfaces, as well as they provide an ability to rapid analysis and data compression for simplifying the data processing that we're obtaining from the sequencing. For the final part of the video, I'll be going over limitations and advantages for the NextSeq sequencing systems. For the limitations, there's a higher error for longer reads compared to other sequencing systems, as well as there's a limited read length of 150 base pairs max and a small flow cell capacity. However, the advantages of the system are there's high throughput, fast turnover times, sample size flexibility, and user-friendly and easy to operate interfaces. Thank you for watching our video.